Hello everyone, welcome to BIS103 lecture 10 and our continued discussion on fat and lipid metabolism. So the topic of this video will be fatty acid biosynthesis, or what to do with excess metabolic fuel. So much like in our discussion about carbohydrates, we first talked about how we can break things down. Now we want to talk about how we can actually make them. So our learning goal for this video will be to be able to describe the reactions and processes required for forming fatty acids and lipids. We mostly will be focusing on fatty acids here, less so on the lipid formation. And we also in this context want to understand the concept of our shuttles again, and here specifically the citrate and malate shuttles. So let's dive right into fatty acid biosynthesis. How can we make fatty acids? How can we make lipids, right? So you can imagine this occurring whenever you have excess fuel. So if you ate a lot of sugars, a lot of other resources, everything that can be broken down into acetyl-CoA actually, you can use to make fatty acids, to make lipids, if you have enough fuel, if acetyl-CoA is not needed for other purposes. This also will help you to deal with your cofactor limitation of CoA. Right? So be aware, right? If you eat too much, all this extra energy will be generated and will be converted into fatty acid into fat. So as I said, right, the major substrate here, talking about acetyl-CoA, is actually acetyl-CoA. So the breakdown product of beta oxidation, so of the catabolism of fatty acids, is in turn the substrate for its biosynthesis. So what we're doing here is that the first thing we do is we're preparing the substrates. So much like what we've seen before for both the breakdown and synthesis of metabolites is we have to activate them. And in fatty acid biosynthesis, it actually is a multi-step process to generate two substrates, right? So let's start here at one. What we're bringing in is our molecule of acetyl-CoA. This now can, for example, and in most cases it will, come from breaking down carbohydrates through glycolysis in the PDH reaction, generating acetyl-CoA. If you do not have demand for generating chemical energy in form of ATP, meaning you would break down the acetyl-CoA through the TCA cycle, you will use this to store it away for later. Right? This acetyl-CoA now we want to activate, and the way we do it is we're actually carboxylating again. Right? This should look familiar. We're using carboxylation for this process. We actually require ATP for this because, like in all of our carboxylation cases that we observe in this course, it is biotin dependent. So we need our cofactor biotin here. Have a look at the video on cofactors if you don't quite remember what biotin looks like and how it works. Right? It's an ATP dependent cofactor here that can bind bicarbonate, so carbon dioxide in solution, and can transfer it to a substrate, in this case acetyl-CoA. What we're generating now is manual CoA. So acetyl-CoA with one additional carbon attached here in form of this carboxylic acid. Again, ATP dependent, we'll see this again later, right? That's important. So making fatty acids, much like we had seen before for gluconeogenesis, requires some ATP to do it. Okay. Now, manual CoA is not quite ready yet. It's not the final substrate for fatty acid synthesis. There actually is another step that's highlighted here under 3. What we're actually doing is we're taking manual CoA now, and we're adding this ACP molecule to it. Right? We'll see in just a second what that actually is. It's a secondary activation step, and it releases CoA. What we're generating is this manual ACP, and this is actually one of our substrates for fatty acid biosynthesis now. Now, under 2 here is our second step. So we're actually bringing in a second molecule of acetyl-CoA, and for this second substrate of fatty acid biosynthesis, we're actually not doing the carboxylation. We are directly activating it by binding it through this ACP. Okay. What we're generating here is this acetyl ACP. Again, we're releasing CoA. So we're releasing some of our cofactor. So our final activated substrates for fatty acid biosynthesis now are this acetyl ACP and the manual ACP. So with all this talk about ACP, let's look at what this actually is. ACP is actually a protein. It's called acyl carrier protein. And if you actually look at its business end right here, and you compare it to our coenzyme A, you can easily see it's very similar. In fact, the functional group, the business end, is actually the same. We have these sulfhydryl groups here that allow us to activate molecules. Okay? So it actually has the same function as our coenzyme A. Both activate these fatty acyl groups in these cases. Just CoA we're using in beta oxidation, and ACP we're using for its biosynthesis. So it's one part of regulating these two opposing pathways by using different activating cofactors. The other reason here is that this ACP, it being a protein, just the way this protein is structured helps for the functional group actually getting access to its substrate, which otherwise would be a bit more difficult. We won't go into the mechanism of this really complex enzyme, but it also has to do with access to the substrate. So now how can we do this? We have generated our activated substrates. How are we making fatty acids? And essentially, you can imagine fatty acid biosynthesis as beta oxidation turned on its head. So we're actually using the same kind of oxidation reactions and reduction reactions to accomplish this. And what we're doing is that we're attaching 2 carbon units to the backbone one at a time using turns of fatty acid synthesis. Much alike what we had done in beta oxidation, where we were releasing 2 carbons at a time in form of acetyl-CoA. Now we're essentially going backwards here. Okay. So in this fatty acid synthase complex, and it's a really huge enzyme complex, in the first round, we're bringing in our 2 substrates, acetyl ACP and manual ACP. The first step, what we're doing is we're actually decarboxylating again, and we're releasing ACP. It sounds counterintuitive, right? Because we just had activated manual ACP by carboxylating this, and it even costed us ATP to do it. But the decarboxylation and the release of ACP here are actually really helpful in this particular reaction to release enough energy to do this and to generate this molecule here that has now double bond trap between 2 carbonyl groups. That should look really familiar from one of the intermediates of beta oxidation. Right? That actually was the endpoint of beta oxidation before we cleave off acetyl-CoA. So it's the exact opposite in essence here in terms of what we're achieving. Okay? So on this side here in blue, this is a part of acetyl ACP that is coming in, and we're releasing the ACP from this substrate. And then from this substrate here, we are releasing the carbon dioxide. And the red part here, the rest of the molecule is now undergoing condensation. With the acetyl part, we're generating this intermediate here. And you do not need to know the names of the intermediates. You just focus on the individual reaction steps here. So now that we have these 2 carbonyl groups here, the next step for us is actually to reduce this carbonyl group right here, okay, into a hydroxy group. As a cofactor, we're using NADPH. Keep in mind, NADPH coming from our pentose phosphate pathway, PPP. It is being oxidized, and this can actually be used for the PPP again as a substrate for the cofactor. Effectively, what we're doing is we're reducing this carbonyl group to a hydroxy group. All right. What's next? Now we want to get rid of this hydroxy group, so we're actually doing a dehydration. We're removing water from this bond here. We're generating a carbon-carbon double bond. Again, this should look very familiar to beta oxidation. And then the last step, again, we're taking this double bond here and we're reducing it by means of oxidizing NADP+. So the electrons are going from NADP to our double bond. It's being reduced to a single bond. We are generating the oxidase cofactor. What we have done now essentially is that we generated an acyl ACP molecule that is 2 carbons longer than what we had up here in our manual ACP. Right? It's a C4 carbon now, right? 1, 2, 3, 4. And this now can go back and undergo another circle of fatty acid biosynthesis. So what is happening now is that you're actually replacing the acetyl ACP as a substrate with the product of your first round of fatty acid biosynthesis, and you're bringing in another molecule of manual ACP. What that also means, though, is that for each of these rounds, you need one ATP to activate to manual ACP. Right? So it's quite energetically demanding to make fatty acids and lipids, but consider how good an energy research and storage component they are. So it's really worthwhile doing that. But by going continuously through this pathway, you can add 2 carbons at a time, and you can do so all the way until you're reaching 16 or 18. Then the hydrocarbon chain becomes too long for the enzyme complex, and it's actually released as a product. So these are the 4 reactions that we're using for these iterative addition of 2 carbon units. Now really compare to beta oxidation, because I think you will really be able to understand both processes if you compare them. Right? If you turn this thing on its head, right, you started in beta oxidation here with, for example, a fully saturated fatty acid. So just imagine this hydrocarbon chain to be longer. We would have gone in the other direction, and we would have first oxidized this double bond to generate a double bond. Then we would have used a hydratase or hydrogen lyase to put water across this double bond, generating a hydroxy group. And the next step would have been an oxidation to the carbonyl group to trap this bond here between the 2 carbonyl groups to cleave it as a 2 carbon unit. Really, fatty acid biosynthesis goes just the other way around, where we start with this double carbonyl bond relationship here. It's a reduction, dehydration, and reduction again. Okay. The other difference is we're using NADPH as a cofactor here instead of NADH, which is what we actually used in a beta oxidation. So just to sort of compare the two in a little bit of a broader picture again, here we have our fatty acid synthesis. Here we have beta oxidation. So we have to compare it to some of the material from the last lecture. Location compartmentalization is different. Fatty acid biosynthesis in animals actually occurs in the cytosol and partially at the endoplasmic reticulum, or ER, whereas beta oxidation occurs in the mitochondria and in some cases in the peroxisome in animals. In plants, it's actually in the chloroplast. We won't look at it, and the ER, and for beta oxidation, the peroxisomes. But beta oxidation and fatty acid synthesis actually do occur in different compartments, which makes a lot of sense considering that you're using some of the same intermediates essentially, but you're moving into opposite directions in terms of pathway directionality. We're also using a different carrier, a different activating group, which is our ACP protein here in the biosynthetic part, whereas beta oxidation uses CoA. As I said, the coenzyme substrate is different. It's NADPH in fatty acid biosynthesis, and it's NAD plus and FAD in beta oxidation. And always keep in mind which is it the reduced or the oxidized cofactors that are using. So have a close look at the pathway directionality. If questions like this come up, you always want to see the correct reduced or oxidized cofactor in this constellation. We actually have a different stereochemistry here, but you can ignore this. We didn't really touch on it. And then very importantly, the ATP requirements are different. In beta oxidation, we needed these 2 ATPs to activate the fatty acid for its breakdown. Here we actually, as I said, we need 1 ATP per every 2 carbons added. So for every round of adding 2 carbon units, we need 1 ATP, which is used for the activation of manual ACP. But that's actually not the entire story. If you look very closely, we actually need 1 more ATP because we need to get the acetyl-CoA into the cytosol in the first place. Right? Look again at the different location of what is happening here. Fatty acid synthesis is in the cytosol. Okay? And we said fatty acid biosynthesis starts with acetyl-CoA, which usually comes from the breakdown of pyruvate through the PDH reaction. Right? You will use fat storage from other resources. So it would come either from sugars or from amino acids. Mostly it will come from sugars. So PDH reaction is happening in the matrix of the mitochondria, right? So in order for fatty acid biosynthesis to even happen, we need to get acetyl-CoA into the cytosol first. That in itself might be possible, but also keep in mind that we discussed several times now that we never want to exchange the cofactor pools across compartments. So you don't just want to get acetyl-CoA over into the cytosol because you want to keep the CoA in place in the matrix. So the way to overcome this is to help ourselves again with one of our metabolite shuttles. And in fact, we're using two here. So what we're actually doing is that we're using one of the reactions of a TCA cycle, and it's a citrate synthase here, the condensation of acetyl-CoA and OAA, right, to citrate. And then we can use a shuttle that we haven't heard of before, the citrate shuttle. They're actually transporters for citrate to go across the membrane of the mitochondria. Citrate can enter the cytosol. And now there is a new enzyme reaction that we also hadn't seen before, where citrate can actually convert it into OAA using a lyase. Right? So what's actually happening is here that you're breaking down your 6-carbon citrate molecule into acetyl-CoA and OAA. That's a 4-carbon molecule and the 2-carbon molecule. So you cleave 2 and 4 carbons away from the 6-carbon molecule. You use this lyase to do so. And this is a reaction that actually requires this one additional ATP. What you have achieved now, right, is that you have acetyl-CoA now in the cytosol, but you haven't used the CoA that was originally here in the acetyl-CoA molecule in the matrix. You actually are bringing in free CoA in this lyase reaction, and you attach it to the enzyme-bound intermediate, the 2-carbon unit that is being cleaved from citrate. CoA will be added. You're generating acetyl-CoA actually de novo here. This now in the cytosol can go into fatty acid biosynthesis. Okay. But you're not quite done yet, right? Because in order for this to cycle efficiently, you also don't want to run out of OAA in your matrix. That would be a problem, right? Because this is a TCA cycle intermediate. So you need to refill it. And if you bring in citrate from the matrix in the cytosol, you're actually actively removing metabolites from the TCA cycle. Now we do have OAA here, right? And now if you remember our lecture of G and how we could do this, we had talked about the malate shuttle. And what we had done there is that we used the reversible interconversion of OAA and malate in this shuttle. Then malate can be transported. This is the exact same shuttle that we use in G. We can bring malate back into the matrix. We have an isozyme there that can do the isomerization in reverse and can refill our OAA that we just used here to make the citrate to bring about the transfer of acetyl-CoA units between the matrix and cytosol. So these shuttles are really critical for making sure we can have pathways that occur in different compartments, cooperate while maintaining the cofactor pools separately in each department. And not only can we use just one shuttle, we can actually combine them with each other to realize these type of transfers. So that concludes our discussion on fatty acid biosynthesis. I will end here. Many of you may have noticed we only ended at sort of the 16 or 18 carbon fatty acids here. If you want to go all the way to the lipids, there are a few more reactions that we need to look at, but they're rather complex and sort of go beyond the scope of BIS103. So if you're very interested in it, we can touch on it in the office hours. You can read up on it also in your textbook.